Welcome back. We're now going to talk about the non-spore forming gram-positive rods. So we're going to look at two. So Corynebacterium diphtheriae and Listeria monocytogenes. Some really nice peritrichous flagella there. I've given you this table for pictures. All right, so Corynebacterium diphtheriae causes the disease diphtheria. This is one that there is a vaccine for, but it was once a very fatal and very common childhood disease. It makes a deadly cytotoxin, and I've given you a little bacteriophage here. So once again, only certain strains of Corynebacterium diphtheriae are going to actually cause the disease. And so through that horizontal mobile genetic pool, there is a gene for a deadly cytotoxin, and so it shuts down protein synthesis, and so that's going to kill cells. So what it causes is a respiratory disease, upper respiratory, so through droplet transmission, and it forms what's called a pseudomembrane. So pseudo means false, it's not really a membrane, but due to those dead cells. And what can happen to this is it can break free and actually get lodged in the airways, or because we have cell death, then the bacteria can travel in the bloodstream. And so they can cause paralysis, it can affect the heart and kidney and cause failure. And we talked about this when we talked about toxins and toxoids and antitoxins. And we all learned about Balto and how that antitoxin was delivered to those children in Nome, Alaska by dog sled. And the toxoid vaccine, clearly that dropped the number of cases of childhood diphtheria, but boosters are required. And so there are some isolated cases because of anti-vaxxers or someone doesn't get their booster. So it has not been eradicated, it still exists. Just because of vaccination, there are very few cases in the United States. This is an exotoxin, just kind of reminding you, we looked at those AB toxins, and so you have the binding portion and then you have the active portion. And so once again, cytotoxins, they shut down protein synthesis in this case and cause cell death. Yeah, once again, so just diphtheria, people need to get their vaccines and also for the boosters. And if they do contract the disease, then we have that antitoxin, so giving antibodies, so antibody therapy. And that's for you. So the other one is Listeria monocytogenes. And we talked about this when we were discussing how bacteria can spread or hide from the immune system. So these are facultative, meaning they have the capability to grow and live inside of our cells, so intracellular parasites. And it can grow within multiple host ranges. It targets the macrophages, so we know if anything gets in a macrophage, these are mobile cells, and it also is going to enter the epithelial cells of the gastrointestinal tract. What it's going to cause, though, because I just said gastrointestinal tract, actually causes meningitis. But remember those little actin tails. We're not going to watch that video again. I just used the slide over again. So I don't know if you remember the sad bluebell recall of 2015. I do love my ice cream. We talked about Listeria monocytogenes in that it is a human pathogen and we're quite warm, but it can grow in 4 degrees centigrade, so in the refrigerator and also in the freezer. So it is a psychotroph. It is a facultative anaerobe, meaning it can grow with or without oxygen, so vacuum packing food isn't going to kill it. So transmission of listeriosis is going to be through ingestion, often of unpasteurized foods, so particularly our group. So epidemiology, it's going to affect pregnant women, and so we get vertical transmission. So pregnant women, that's why they are advised to not eat soft cheeses or unpasteurized foods. Also, cold foods, so foods that take preparation, or cold deli meats, or like hot dogs if you don't cook them. So this is a case where cooking food is going to get rid of the organism, but often, like I said, these are going to be foods that are not cooked, so chilled foods. So in most people, you can ingest this and it's going to be asymptomatic, so nothing's going to happen. If someone is immunocompromised or if a woman is pregnant, this can cross the placenta. So not very many things can, but this is a case of vertical transmission. So it infects the fetus in utero. Then it can cause miscarriage or fetal death, but more often the baby is delivered about 1 to 4 weeks later because of this organism being able to travel through cells and also live inside macrophages. It will travel to the coverings of the brain and the spinal cord, so the meninges. So about 1 to 4 weeks later, it causes neonatal meningitis, and it is about 1/3 of the time fatal. So this infects the immunocompromised. It can also cause meningitis. Pregnant women, typically the woman is going to be fine, but then it crosses the placenta, affects the fetus. I'm just kind of showing you that mechanism of pathogenesis. They can grow inside of cells. They can grow inside of a phagosome, so they can live inside macrophages, and they're mobile cells. They can also use their actin tails to spread. So they enter through the epithelium of the gastrointestinal tract. And then through those actin tails, they can be released. And so we get spread and protection from the immune system because of their ability to live inside cells and travel from cell to cell. Yeah, that's for you. So now I have the acid-fast bacterium, so the genre Mycobacterium, and we have 2 diseases, so Mycobacterium tuberculosis and Mycobacterium leprae. So as a genus, remember we've kind of talked about them already. They have a unique cell wall. They're made up of mycolic acid, so very, very thick. They are acid fast, so that means there's a special staining used to identify them. They can resist decolorization with an acid alcohol. This also makes them very resistant to disinfectants. They are aerobic, so they need air. They are pleomorphic, meaning they change shape. They're technically gram-positive, but because they're mycolic acids, for detection and diagnosis, you would do a special stain, an acid-fast stain. They too can grow inside of cells, so intracellular parasites. What's unique about them, because their cell walls are very large, that mycolic acid is a huge molecule. They have a very long generation time, like hours. It takes them hours to divide, so they're very slow growing. So as far as incubation period of disease, you could be exposed and not have any disease symptoms for years. Very slow growing, very difficult to grow. Often, I worked at LSU at the vet school, and part of the building I was in was part of the Hansen's Disease Center. And so we had a colony of armadillos, and so they would grow Mycobacterium leprae in the foot pads of armadillos. It is also endemic, meaning it's not going to go away because armadillos carry this bacteria. Humans are the only natural reservoir, however, for Mycobacterium tuberculosis, so the causative agent of tuberculosis. Because these organisms are so slow growing, usually most people's immune systems will handle any sort of exposure. Once again, your populations at risk are going to be the elderly, children. Throw in also immunocompromised. What does that to poverty, poor nutrition, poor sanitation is going to come into play here. Yep, I'm going to do it this way. So let's learn about tuberculosis first.
In 2008, archaeologists uncovered 2 9,000-year-old skeletons. There's no definitive way of knowing what killed these ancient people, but we do know their bones were infected by an all-too-familiar bacterium. The ancient Greeks knew its consumptive effects as phthisis. The Incans called it chaki-onke, and the English called it tuberculosis. Today, tuberculosis, or TB, is still one of the world's biggest infectious killers, causing more deaths than malaria or even HIV and AIDS. But what exactly is this disease, and how has this pathogen persisted for so long? Typically, TB bacteria, called Mycobacterium tuberculosis, are airborne. They travel into our airways and infect our lungs. Here, immune cells called macrophages rush to the infection site, attempting to absorb and break down the bacterial invaders. In many cases, this response is enough to remove the bacteria, but in individuals with other medical conditions, ranging from malnutrition and HIV to diabetes and pregnancy, the immune response may not be strong enough to destroy the intruder. If so, Mycobacterium tuberculosis will reproduce inside those macrophages and form colonies in the surrounding lung tissue. As they infect more cells, the bacteria employ cell-degrading enzymes that destroy the infected tissue, triggering chest pain and causing patients to cough up blood. The damage to the lungs leads to oxygen deprivation. This begins a flood of hormonal changes, including a decrease in appetite and iron production. From here, microbes can spread to the skeletal system, causing back pain and difficulty moving, to the kidneys and intestines causing abdominal pain, and to the brain, causing headaches and even impaired consciousness. These symptoms produce the classic image of TB: weight loss, a hacking bloody cough, and ashen skin. This ghostly appearance earned TB the title of the White Plague in Victorian-era England. During this period, tuberculosis was considered a romantic disease because it tended to affect poverty-stricken artists and poets, those with weaker immune systems. TB's outward symptoms even helped fuel the popular myth of vampirism. In spite of, or perhaps because of, these less than scientific concerns, this period also marked the first strides toward curing TB. In 1882, the German physician Robert Koch identified the disease's bacterial origins. 13 years later, physicist Wilhelm Röntgen discovered the X-ray, enabling physicians to diagnose and track its progression through the body. These techniques allowed researchers to develop reliable and effective vaccines, first for smallpox and again in 1921, when scientists developed the BCG vaccine to battle TB. These developments laid the groundwork for the modern field of antibiotics, currently home to our most effective TB treatments. But antibiotics fail to address a major diagnostic complication. About 90% of people infected with TB don't show any symptoms. In these latent infections, the TB bacterium may be dormant, only activating when someone's immune system is too weak to mount a defense. This makes TB much harder to diagnose, and even when properly identified, traditional treatments can take up to 9 months, requiring multiple drugs and a high potential for side effects. This discourages people from finishing the full course, and partial treatment enables bacteria to develop resistance to these drugs. Today, the disease is still prevalent in 30 countries, most of which face other health crises that exacerbate TB and trigger latent cases. Worse still, accessing treatment can be difficult in many of these countries, and the stigma towards TB can discourage people from getting the help they need. Health experts agree we need to develop better diagnostics, faster-acting antibiotics, and more effective vaccines. Researchers have already developed a urine test that yields results in 12 hours, as well as a new oral treatment that could cut treatment time by 75%. Hopefully, with advancements like these, we'll finally be able to make TB exclusively a thing of the past.
Okay, so once again, another reason to be grateful you live in the United States. Yeah, so there is multi-drug resistant tuberculosis now, 500,000 new cases in 2015, and now there is extra drug resistant tuberculosis, meaning there's very few treatment options for them, so pretty much 70% mortality within months of diagnosis for those. So how it works, and just to kind of flesh out, that was a great video, that's why I used it. So people are exposed to Mycobacterium tuberculosis, and only those that have risk factors will it not be cleared, because once again, Mycobacterium tuberculosis, it's so slow growing, the immune system is going to remove it. So once someone is exposed, they have what's called a latent infection, and remember, if you see latent, it means you have to wait. So latent tuberculosis infection. So they're asymptomatic, or they're just going to have a mild lung infection. So these people aren't infectious, but they are walking around with the bacteria. So what happens then later in life, once again, they become immunocompromised. Like I said, a lot of this is going to be due to poverty, starvation, and then 2 people living in very close quarters, once you get the actual tuberculosis disease, and it kind of showed the symptoms of that, so they used to call it consumption, so weight loss, night sweats, they cough, and through respiratory droplets, it spreads. So usually, like I said, it's going to be when people live in poor poverty conditions and in close quarters. Actually, my grandfather and my aunt died of TB during World War II. So there usually are these risk factors that are going to set people up for developing the disease. I think the video did kind of a nice job. Like I said, the macrophages, they can't get rid of them because of that waxy wall, and so you get what are called gone foci. So they just build a wall around it, and so they start taking up places in the lungs. And so that's what the video was talking about with the invention of the X-ray machine, that then they could sort of watch the progression of tuberculosis. And then you can see that histologically on a slide. And so it could stay encased, but then when someone gets the disease, then they break free, and then it's no longer latent. And so then you'd have the symptoms. So exposure, late infection, and then the actual disease, TB, and that's when coughing up the sputum. And then you could look for the bacterium doing an acid-fast stain. The second disease caused by the acid-fast bacteria is Hansen's disease, or leprosy. Hansen's disease is more politically correct and less stigmatized. There was a man named Gerhard Hansen, and like I said, he set up a research facility in Louisiana, and so he became that leading authority on that bacterium. And like I said, it was the first one linked to the particular disease by Robert Koch. And yep, LSU. That's where I was, right there. It is, once again, it's still a huge problem in other parts of the world for the same reasons. So just poor sanitation, just poor health overall, and people living in close quarters. Like I said, it's never going to go away. It is endemic in armadillos, and it can be spread to humans, and then through human-to-human contact. It has a very long incubation period, like I said, 3 years. And most people are exposed and take care of it, just like in tuberculosis. But it is the one disease, though, that's going to affect peripheral nerves. And so loss of sensitization, and then two, also it prefers cooler temperatures, so usually on the appendages. So tips of the nose, fingertips can be lost. So our next up are the wall-less bacteria. So the genus Mycoplasma, and we're looking at Mycoplasma pneumoniae, so causes of pneumonia. They are the smallest of the free-living bacteria, so not obligate intracellular parasites. They are less than a micrometer long. They're called wall-less because they don't have any peptidoglycan. Instead, they have an extra cell membrane. Since they don't have a cell wall, clearly, beta-lactams or penicillins cannot work on them, so that type of antibiotic cannot work. Also, though, because they have this cell wall, they're not going to really have a shape, so they don't have a defined shape, so they're pleomorphic. They look like little fried eggs on an agar plate. And the other thing about them, because they don't have a peptidoglycan cell wall, they cannot live for very long out of a body, and so they're going to be through close contact transmitted from person to person. The type of pneumonia Mycoplasma pneumoniae causes is called an atypical or walking pneumonia. It is atypical because instead of causing a wet cough, it causes a dry cough. It is very slow to develop, so 1 to 3 weeks. It is transmitted through respiratory droplets. And once again, this is not going to be through fomites, so someone has to be close to you. So really, when you see these breakouts of this type of pneumonia, someone is living in the dormitory, they're in the army, so it seems to rip through barracks, or they are in prison. And what this does is there's a cytotoxic protein that is going to damage those cilia, and because the cilia can't row, that organism then can colonize those lower respiratory airways. And it's not causing mucus production alveoli, it's damaging the lung tissue around the alveoli. So like I said, you get that dry cough. And so whereas Streptococcus pneumoniae affects older people, this one you find younger people, like I said, usually living in close contact in community. There is treatment, antibiotics, you just can't use those beta-lactams because they don't have a cell wall. There is no vaccine offered for it. You just have to sort of avoid crowds if you are worried about contracting it. Its method of pathogenesis, it has adhesin proteins which allow it to adhere to the respiratory epithelium. It has toxic proteins and it releases those reactive oxidative species, so hydrogen peroxide, and it damages those cilia, so therefore they can colonize the lower respiratory tract. And then they also have super antigen activity that causes that inflammation around the alveoli, so dry cough, headache, low-grade fever, and just general overall feeling crappy, malaise. Like I said, usually people are walking around with this, so that's why they call it walking pneumonia. And then too, that can also be seen on an x-ray. So that's why when somebody is coughing, they often do x-rays because you can see the grand foci. For tuberculosis, you can see it, and then pneumonia shows up as that whitening on an x-ray. So it starts off with cold-like symptoms. No sputum. So yeah, just comparing the two. So yeah, I call this one the community-acquired pneumonia of the elderly. Also causes other things. We talked about meningitis and ear infections, and this is sort of that community, the crowded young, so young peeps, to keep those two straight. So Mycoplasma pneumoniae, Wallace, Streptococcus pneumoniae, pneumococcus. All right, I'm going to stop there. Last one, come back next time for the spirochetes.